Non-equilibrium noise emerging from broken detailed balance in active gels. This paper investigates how non-equilibrium fluctuations, also called active noise, arise in active gels, which are materials composed of elastic filaments interconnected by transient molecular motors or crosslinkers. These active gels serve as simplified models of cellular structures such as the cell cortex and mitotic spindle, where mechanical properties and dynamic behaviors are critically influenced by molecular activity. Understanding the origin and characteristics of the non-equilibrium noise in these active gels is important because it governs force generation and mechanical response in living cells, with potential implications for biomimetic materials and understanding cellular processes. The authors approach this problem by modeling the gel as a network of elastic elements connected by crosslinkers that can bind and unbind stochastically. Each crosslinker exists in bound states, characterized by a mesoscopic strain and an orientation, which together define the state of the linker within the network. Molecular activity manifests as a departure from detailed balance, quantified by an activity parameter that modulates binding and unbinding rates, and thus drives the system out of equilibrium. This framework allows the authors to connect microscopic molecular kinetics with macroscopic mechanical properties and fluctuations, providing insights into how non-equilibrium noise emerges from broken detailed balance in active gels. Figure 1 illustrates the physical system and the key components of the model. Panel A depicts biological contexts where this active gel model applies, including the cell cortex and mitotic spindle, which consist of networks of cytoskeletal filaments interconnected by active molecular motors and crosslinkers. Panel B zooms in on a coarse-grained volume that contains the active gel. Within this volume, the filament network establishes a nematic order, represented by the tensor Q, and the system may be subjected to an external shear rate, V. Each bound molecular motor or crosslinker is described by two variables, a mesoscopic strain U, which characterizes its elastic deformation, and an orientation Q that denotes the directionality of the motor. Importantly, each motor's strain and orientation are independent of others. Panel C presents the energy landscape of a crosslinker in its bound state. For simplicity, the illustration sets the activity parameter to 0, reflecting thermal equilibrium conditions. In this model, the activation energy required for unbinding remains constant across all bound states, which implies the unbinding rate Ku is independent of the motor's strain or orientation. This is a good approximation when the chemical binding energy dominates over mechanical contributions. Together, these panels establish the central features of the theoretical framework for understanding the dynamics and non-equilibrium behavior of active gels based on molecular scale processes. Figure 2 explores how molecular activity and external driving alter the probability distribution of the stress tensor component, specifically U03C311. Panel A shows the distribution at thermal equilibrium with no molecular activity, where the distribution is symmetric about 0 and the mean stress vanishes. This isotropic case serves as a baseline for comparison. Panel B introduces molecular activity, breaking detailed balance and resulting in asymmetry in the stress distribution. The authors consider a specific form of molecular activity that promotes binding at a preferred orientation, here denoted as Q equals the nematic order parameter Q, with Q11 equals 1. Due to a positive elastonematic coupling, the crosslinkers are biased to bind in states with positive strain component U11. As stress depends on both U and Q, this bias translates to stress being skewed towards positive values, yielding a positive mean stress. Panel C further includes an external shear with a positive shear rate V11, which shifts the stress distribution in the direction of the applied shear. Although molecular activity and external driving additively affect the average stress, their combined impact on the steady-state distribution is not simply additive, due to their non-commuting effects on linker states. This results in cross-coupling terms influencing the variance of the stress tensor and hence the noise amplitude. Thus, Figure 2 elucidates how active fluctuations arising from broken detailed balance and external driving jointly shape the mechanical fluctuations in active gels. Figure 3 presents the response function and fluctuation spectrum of the system, demonstrating how molecular activity breaks equilibrium conditions. The response function, K of omega, characterizes how the system reacts to external forces at frequency omega, while the fluctuation spectrum, SR of omega, measures spontaneous fluctuations at these frequencies. Under thermal equilibrium conditions, when detailed balance holds and molecular activity parameter gamma equals 0, the fluctuation dissipation theorem, FDT, applies, implying that fluctuations and response are directly related. This equilibrium case is depicted by the dashed line in Figure 3, where the fluctuation spectrum and response function coincide. When activity is present and gamma is non-zero, this symmetry breaks down. The scaled fluctuation spectrum deviates from the response function, highlighting non-equilibrium behavior, where fluctuations are no longer entirely dictated by dissipation. Moreover, because of the nematic ordering characterized by Q, the spectrum becomes anisotropic, differing along directions parallel and perpendicular to the order axis. Thus, this figure illustrates a generalized fluctuation-dissipation activity relation, quantitatively capturing how activity-induced breaking of detailed balance modifies the frequency-dependent mechanical response and spontaneous fluctuations in active gels. Figure 4 illustrates the concept of Ornstein-Uhlenbeck colored noise, characterized by a time-correlated autocorrelation function rather than the memoryless white noise. The figure emphasizes that for colored noise, the conditional expectation of the noise at a later time, given its value at an earlier time, does not vanish, unlike in white noise. This implies that the simple exponential decay formula for the evolution of a stochastic variable under white noise does not apply here. This depiction helps in understanding the complexities of modeling active noise, which often exhibits temporal correlations reflecting the underlying molecular kinetics and fluctuating forces in active gels. Figure 5 visualizes the steady-state distribution of the stress tensor components produced by bound molecular linkers within 2 dimensions. The 2 independent components chosen for the axes are 03C311 and 03C312. The figure also features black streamlines that indicate the orientation of the principal axes of the stress tensor at each point, directions along which shear stress vanishes. For example, a point at 0,1 corresponds to a stress tensor with principal axes oriented at 45 degrees, which is reflected by the tangent of the streamline at that point. Bright spots on the plots highlight regions with high probability density, indicating the most probable stress states under the influence of activity. Similar to Figure 2, the activity parameter is set to promote binding in states with a preferred direction, affecting the shape and orientation of the stress distribution. This detailed 2-dimensional representation conveys the complexity and anatoprsy of the stress fluctuations arising from molecular activity in active gels, providing a richer picture than 1-dimensional projections alone. Figure 6 illustrates the abstract state space of a single molecular linker, modeled as a continuous time Markov chain. The system consists of an unbound state, represented by the state variable mt being 0 everywhere, and an infinite set of bound states, parameterized continuously by position or strain coordinate x0. When the linker binds to a particular state x0 at time t, mt becomes a Dirac delta function peaked at x0, indicating localization in state space. The linker transitions between these states according to binding rates kbx that depend on the target state and a constant unbinding rate ku, independent of state. Because a linker can only bind when unbound, state transitions proceed via unbinding from the current state and binding to a new state. Assuming independence among linkers, this Markov chain description underpins the macroscopic formulation of the fraction of bound linkers and total force in the system. This representation connects stochastic molecular kinetics with emergent mechanical properties at larger scales. In conclusion, this study elucidates how non-equilibrium noise and active fluctuations emerge in active gels as a direct consequence of broken detailed balance at the molecular level. By modeling the cytoskeleton-inspired gel as a network of elastic filaments connected by transient molecular motors characterized by mesoscopic strain and orientation, the authors connect microscopic binding kinetics with macroscopic mechanical stress and fluctuations. The molecular activity parameter quantifies the departure from equilibrium, modifying the distribution of bound linker states and thus shifting and skewing the stress probability distribution. External driving such as shear flow further influence stress statistics, contributing non-trivially to the noise amplitude due to non-commuting effects on linker populations. frequency-dependent response and fluctuation spectra reveal clear departures from the classical fluctuation dissipation theorem, illustrating anisotropic and active-driven mechanical fluctuations in gels. Altogether, the framework provides a comprehensive physical basis for understanding the rich non-equilibrium behaviors of active gels observed in cellular contexts and synthetic biomimetic materials, highlighting the central role of molecular scale detail, breaking detailed balance in driving emergent active noise and mechanics.